Ladies and gentlemen, welcome to Compass, to Quotidiano TV today. A wonderful good afternoon to Princeton, to Professor Creager, Angela Creager, and a wonderful good evening to Bangalore, to Professor Neeraja Sankaran. I hope I pronounced correctly your name. Welcome to our today's interview about the history of the research of the tobacco mosaic virus. Thanks for joining me. Thanks for taking your time to discuss about this very interesting virus, which is absolutely fascinating. And I hope to also to gain the attention and the interest in this topic now also from our audience. Thanks for joining me.
Thank you.
I'd like, first of all, to introduce Professor Creager. Angela Creager studies the history of the 20th century biomedical research. Professor Creager graduated from Rice University with a double major in biochemistry in English back to 1985 and completed a PhD in biochemistry at the University of California, Berkeley, where she developed also an interest in the history of biology. Now, supported by postdoctoral awards, she retained as a historian of science at Harvard University and MIT, and joined Princeton History Department 1994. So her first book, The Life of a Virus, the Tobacco Mosaic Virus as an Experimental Model, 1930-1965, published 2002, shows actually how a virus that attacks the tobacco plants came to play a central role in the development of virology and molecular biology. So you're actually also the author of several other scientific publications, but in our today's interview, we'll stay with the history of the research of the tobacco mosaic virus.
Great.
Dr. Neeraja Sankaran trained as a microbiologist and science writer before obtaining her PhD in the history of science and medicine at Yale University, with the thesis McFarlane Burnett and the Nature of the Bacteriophage, 1924 to 1937. Currently, she works as an independent scholar at the National Center for Biological Science in Bangalore, India. Neeraja's tale actually begins in about the second decade of the 20th century with the discovery of bacteriophage and the RSV, and extends forward by about half a century. And her contribution is to draw actually parallels between the tales of her two protagonists, the bacteriophage and the rose sarcoma virus. So a tale of two viruses. So parallels in the research trajectories of tumor and bacterial viruses, which was published 2021 and had a profound echo in the world of science. But today, dealing with our today's interview, in your case, Neeraja, it's about the tobacco, the history of the tobacco virus, and here you will cover and we'll look at the research on another paper you wrote on the historical significance of Bernick and his concept of the contagium vivum fluidum for modern virology. I hope I did not leave too much out from presenting both of you with a very impressive career path. I'd like to know from you, what have inspired both of you to investigate this virus? Because the work of both of you shows a lot of fascination and passion for this subject.
Angela, please go first.
Okay. So I had, as you've mentioned, a background in biochemistry. And as I began to get interested in the history of biochemistry and molecular biology, especially as they were taking shape after World War II, when molecular biology became more of a university discipline, I was struck that many of the early accounts of what is often called the origins of molecular biology point to bacteriophage as a really important virus. That was a virus that was studied by Max Delbruck, Salvador Luria, and Alfred Hershey, and for which they received the 1968 Nobel Prize, I think. I'd have to make sure I have the date right. But they trained a whole generation of scientists, some from the physical sciences, to commit to studying a particular kind of bacteriophage. And with everybody working on the same bacterial virus, they could compare results, exchange mutant strains, and have the same sort of collective knowledge building that had earlier exist with model organisms like Drosophila, for instance, the fruit fly, which became a really important model organism in the early 20th century for genetics. What was striking to me doing my PhD at Berkeley in Wendell Stanley Hall was that I was aware that there was another virus that was also being very widely studied after World War II by biochemists and molecular biologists, and it was Tobacco Mosaic virus. And in effect, bacteriophage was used more to try to get at virus genetics, and Tobacco Mosaic virus was often used to get more at biochemistry or biophysics. And the reason for this is actually kind of material, in that if you grow Tobacco Mosaic virus in tobacco plants, you get a very high concentration of virus particles in the tobacco sap. It is then quite a straightforward procedure to chemically purify that Tobacco Mosaic virus particle from infected tobacco plants, and you can get a lot of it, and you can do chemical experiments and other kinds of biophysical characterizations. With bacteriophage, it's actually much more difficult to isolate it biochemically, although I think, it could be done, but not as early. Whereas the way in which these bacteriophages, when they're in bacterial cells, recombine with one another, opened up bacteriophage to the same kind of genetic analysis that had been so successfully done with Drosophila. So these seem to me to be very interesting comparative cases, each of which shows how certain earlier developments in biology fed into molecular biology, genetics on the one hand through virus genetics, and biochemistry on the other hand through virus biochemistry. So for me, I was at first interested in understanding Wendell Stanley's role in molecular biology, because he'd been often dismissed a bit because he came from the Midwest, whereas Max Delbruck was from Germany and was extremely cosmopolitan. I wanted to try to figure out what Stanley's contribution was, and he founded a virus laboratory at University of California, Berkeley, that was quite influential, in which people there were also studying phage, including some of the people in Delbruck and Luria's phage group. And gradually I realized that what was interesting about the story was less Stanley, although he plays an important role in my work, but the Tobacco Mosaic virus itself as an object of study. Having been the first virus discovered independently in 1892 and in 1898 by Ivanowski in Russia and Beirink in the Netherlands, it continued to be studied through the early 20th century. And when Stanley picked it up as a postdoc at Princeton, but not at the university, the Rockefeller Institute for Medical Research, now Rockefeller University, had a plant and animal pathology laboratory not far from this, from where the university is, kind of across what is now the Route 1 highway. And the reason that lab was there instead of in New York with the rest of the Rockefeller Institute was that they needed kind of a rural landscape for all the animals and plants. And it was there that he worked on Tobacco Mosaic virus and was able to obtain it in such high purity that it actually precipitated as these long fibrous crystals. They're actually not three-dimensional crystals, but two-dimensional crystals, because TMV is a very long
come to that,
spiral rod. Yeah. But part of what I found is that crystallization procedure, although kind of sensational at the time, very quickly gave way to using the ultracentrifuge as a better way to isolate these long particles and study them.
We'll come to Stanley. Sorry for interrupting. I was just curious to know what determined Dr. Sankaran to investigate this Tobacco Mosaic virus. And please tell us how you got to know Professor Creager. I mean, that's also part of this discussion. I mean, the two of you also have something in common, except in addition to investigating the history and in the research of the Tobacco Mosaic virus, your academic careers had a very special encounter.
Several, actually, since Princeton was the first university I visited when I started to try and make the move from being a science writer to being a historian. And Angela gave me my thesis topic when she mentioned in a visit to Yale, she mentioned, Joshua Lederberg says that Macfarlane Burnett's work on bacteriophage has not been studied by anybody. And it sat in my brain for a couple of years. And then a few years when I got around to figuring out what my topic was going to be, that's what I decided to home in on. My encounter with tobacco mosaic virus, other than reading Angela's book early in my PhD, when I was working towards my oral comprehensives, was as background as for the theoretical, what it brought to. So I'm fumbling a bit, but please excuse me. Let me back up a bit. So once I got my bacteriophage idea, it came in that phase when Burnett worked on it was in the 20s and 30s, a bit before Delbruck and others picked it up and made it the famous vehicle for molecular biology. And before I'd ever come to starting graduate school, I'd worked on a kind of my life as a proto-historian. I'd worked at Rockefeller University as a science writer, investigating different kinds of things that their scientists had done. And one of the things that I was working on was somebody, in a way, who became a sort of successor to Stanley in that he made the same jump from Rockefeller to Berkeley and very recently moved to Vanderbilt. This is John Kurian. But that's when I was introduced to the RSV, the 1990s iteration of the sarcoma and what they were doing with it. And they said, well, this has the oldest history because the history of this virus at our institute goes back to 1911. It was discovered here. And now we are trying to figure out, and now we are almost figuring out how it causes cancer. So when I was going back over when I started my PhD and worked on it, it occurred to me that there were lots of similarities, parallels in the way the discoverers had conceived of bacteriophage, had conceived of the cancer virus, and that all of it hinged on something called lysogeny. The resolution hinged on something called lysogeny. When I proposed that as a book, when it was taken up as an idea, one person said, well, but you need to go back to, the backdrop for all of this was what was a virus? The virus itself had changed in meaning. When were viruses discovered and what did it mean to be a virus? And in order to get at that, I had to visit the first virus that was discovered, which was Tobacco Mosaic Virus. And so it was deep background for the history I was trying to tell in my book that took me back to the Tobacco Mosaic Virus. And the Beyering paper is what I call a spin-off paper because it was just so compelling to me exactly what Beyering had found and how his idea was brought into the 20th century and beyond, and how it was tethering, the scientists in the 20th century had tethered themselves to Beyering's idea in order to create the discipline of virology that I had to write a paper on it because I didn't have enough room in my book to really mention more than a few paragraphs. And so that became I took some time to write that paper, and it proved to be really rewarding and discovering what all. And so mine, where Angela goes a lot into material, the materiality in my work comes much later, I think. The early part is very much how people thought about these things, thought about things they couldn't see. Yeah. So that's where I got into that's where I got into the Tobacco Mosaic Virus, and its story is part of chapter 3 and in my book.
I think the idea to look at the concept of the research is like, if I may compare it, and please correct me if I'm wrong what we're saying that, one thing is to build a car and the other thing is city planning. You may have thousands of cars without a proper city planning and infrastructure. These cars won't won't be able to travel. People won't be able to use them. So scientists make discoveries, but I have the impression that Professor Creager's contribution is as valuable as the discovery, because you put a discovery into a concept and extrapolate from that concept and say, look, it may be applied and used in order for better understanding or to promote or extend scientific research in other areas, starting with a model. Is the comparison which I made a proper one, also with the idea, motivated by the idea to provide a better understanding for the audience, why a researcher deals with the concept and with the model of a research?
I think that's very well put, actually. And the point is that in order to make research relevant to other kinds of problems, you need to have a way to connect the object you're studying and your ideas about it to other similar objects, especially objects that may affect humans. And ultimately, much of the motivation for study of viruses in plants and animals is to understand those viruses that afflict humans. Although, obviously, tobacco itself was an important commodity, and some animal viruses as well are studied because for livestock, one needs to be able to actually treat or prevent these viral infections. So it's precisely this need for comparison that drew me to think about how something serves as a model system. In other words, it's a model for something else. And here, if I can just use a few things. First of all, I wanted to pull up Neeraja's book on RSV and phage, which is really great. And I have my book, but I don't have the book that has the cover on it. This is just the hard copy, but this is my book on TMV. And here you see an electron micrograph of TMV showing these long rod-shaped particles. What is of interest to me in the history of virology, and what Dr. Sankaran does so well as well, is to show how connections and analogies are drawn between these different viruses, and how seeing how one virus behaves in a certain way can lead you to see how another virus might behave in that similar way. It might lead you to solve a puzzle in studying a different virus, especially a virus in a more complex system. And I think it's the interplay of information between TMV, RSV, and phage. Those are really, I think, in the first half of the 20th century, the kind of leading exemplary viruses for researchers. RSV being a little bit more biomedical, phage being turned into more of a genetic object, and TMV being more biochemical. The interplay between them, the ability to draw analogies, can in turn kind of inspire new kinds of experiments, or even new kinds of ways to try to visualize or to represent or to intervene with the virus system that you're working with. Like you try a new tool, or you try a new manner of plating it in a cell culture because you're inspired by work on other viruses. So that's where I feel like our work is very deeply in conversation with each other. And there have been scholars in the last 10 years that have thought a lot about how this so-called model systems approach to biology, where you study a handful of things very, very, very well. There's a great visual for this on the cover of a book that I edited with a few other people on, it's called Science Without Laws, Model Systems Cases, Exemplary Narratives. And this is an image that was on the journal American Scientist. That is an artist's representation of the leading model systems in biology. It's
I get the zebrafish.
Yeah, zebrafish, fly, mouse, what's the mustard weed? That was the name of the mustard weed. It'll come back to me. And yeast. And these were kind of the leading exemplary materials. And in fact, the Human Genome Project was not just about the human genome. It was about sequencing all of these genomes, where you could actually do experiments. So I think some people have drawn attention to the fact that this kind of work carries with it comparison, which is kind of like older natural historical understandings of biology. And that comparison and experiment kind of are always running together. And I think for me, the notion of a model system is a way to combine them. It's about experimentation, but also comparison.
It's a tool. I mean, I only hold my PhD in economics, and I must say that models are basics economics. You cannot teach economics, you cannot understand economics without models. So this type of concept seems to be something universal, and particular thing for researchers in the area of chemistry and biology. There is a need for other scientists to communicate it and to offer them a model in order to let more people benefit from this research and have the results of research proliferate to the benefit of either the health or the prosperity of humans. And I've observed that, I mean, what you two also have in common is that looking, Dr. Sankaran, at the concept of contagium vivum fluidum, there's again a concept which at a certain extent revolutionized science. It's again, he had again a theory. If you could please explain us what actually Bernick did, his role in science, and at which extent he also was dealing with a model and a concept.
So Byrink was trained, first of all, as a botanist. And he was in the laboratory, or he was a colleague of the very first person who we know, who scientifically investigated the mosaic disease. The tobacco mosaic disease, and I won't call it a virus yet because the word virus was not yet around. I mean, the concept of a virus causing a disease came about. I think tobacco mosaic virus is the first. It's Frankel Conrad, wasn't it, who called it almost always the first virus in pretty much all areas of virology. But Byrink was there and at first was very interested in what Adolf Meyer was doing. Adolf Meyer investigated it and first showed that this was a transmissible disease in much the same fashion that bacterial diseases, this is in the late 19th century. So the germ theory is just getting established, and he was trained in that tradition of Koch. And Byrink was not, but then he trained himself. So he was interested, but then he could not take it any further. And about the time that Meyer's paper came out in 1870, a few years before, it's in the 1880s. I can't get the exact date. A few years before, he got a new job and moved to Delft, and subsequently picked up and started investigating this disease by himself. And what he found is, by then filters had improved, and he found that this this disease-causing principle that was in the liquid was filterable. It could pass through the filters. And he did a series of very detailed experiments in which he separated layers of diffusion of this liquid trickling down through agar and showed that each layer was still infective. And so he believed that what he had was a liquid or a soluble contagium. And he says this is not a contagium fixum, that is not particulate, but that it is soluble or dissolvable or liquid. I mean, these are words that are being used interchangeably. And one more extremely important thing, he said that the multiplication of this is intimately tied with the multiplication of the host, of the disease, of the plant in which it's causing the organism. And this is what, so he thought this is very different. The disease principle is different than the other kinds of diseases that we're talking about. His idea was far too outlandish in his time for people to be able to even grasp, much less test. And therefore it was Ivanovsky who had discovered, who was the first person to show that the agent of the tobacco mosaic disease was filterable a few years before Byrink started his work, could not accept what Byrink said. And there was this massive difference of ideas conceptually. And it wasn't until decades later, really, after more was known about how viruses were different after viruses could be seen and grown in other things. So what Baerink was talking about was something by which we define viruses today, which is their obligate parasitism. They have to live in, they have to have a living host cell in order to be able to show signs of life, in order to do their stuff, so to speak. And that was the concept then that, you know, people who started to call virology a discipline when it came into its own. Until then, you know, the first several decades of the 20th century, virology was very much, viruses were considered to be small bacteria. And, and therefore virology was just a child of, you know, bacteriology. But as these differences began to become clear, virologists became a separate discipline. Yeah. And that, I mean, some of the youngest or the frontier pioneers of that discipline would be Tom Rivers. And he's the one who then looks back and says, well, I consider Baerink to be the father of our discipline. There are others who consider Ivanovsky. And the reason I consider Baerink to be the father is he saw this agent as being different. He saw it as a distinct entity. And of course, it was several more decades, but that was Baerink's idea. So he gave us effectively in very, very, you know, 19th century terms and terminology, and with the lack of experiments, came up with this idea that could not be tested until many, many decades later. And but also through different other things that they found, and until various material properties of viruses were found out. Viruses, you know, Stanley, as Angela mentioned, crystallized the virus, and then the electron microscope came along. And at that time, many viruses were simultaneously. I mean, if you look at the difference at when different viruses were discovered, either due to their effects or because of seeing them, mostly, well, due to their effects in plants and bacteria, in animals, it begins in the 19th century, and keeps going on. I mean, we're still finding viruses today, right? 150 years later. But if you look at the electron microscopy, they were all found 1 year after the other. Bacteria, boom, boom, boom. Once you figured out the electron microscope, they just started putting everything under to start looking at it. And you could see RSV still remained elusive, the Rouse sarcoma virus, but for other reasons. But I'm digressing. So yeah, so Bayerink's main contribution was a theoretical one on the conceptual of virus.
May I build on this with a few comments? I really admire Dr. Sankaran's analysis of Bayerink's early conceptualization. It's just spot on. I was so happy to be able to cite it in a recent piece that I wrote for Annual Review of Virology.
Thank you.
What's interesting, Ivanovsky in 1892, this Russian bacteriologist, he couldn't visualize Tobacco Mosaic virus in a microscope, and he couldn't culture it. And he couldn't capture it on a bacterial filter, but he still thought it's some sort of a tiny bacteria. It's just smaller. But then when Baerink was also identifying it 6 years later, he thought there's something about this that is different. I mean, as Dr. Sankaran said, and what's striking is that their dispute over whether this is just a small bacteria or something distinctive would have not had any importance if it were not for the discovery of other viruses in the decade after TMV. So this is the first one found, but within 10, 11 years of Ivanovsky's original publication, viruses were identified to be the agents of foot and mouth disease, rabbit myxomatosis, African horse sickness, foul plague, yellow fever, and rabies. Many of these are dangerous animal and human viruses. So suddenly, what looks like a very philosophical question over whether something is a solution or a particle becomes very important. And for quite some time, the definition of virus was a negative definition. It
like
goes
poison.
through a bacterial filter, right? It can't be filtered, it can't be visualized in a microscope, and it can't be cultured. So how do you advance a field when the main attributes of the subject are defined negatively? And this is where the techniques of biochemistry, biophysics, genetics, and other fields become really important to find some way to pin down these elusive viruses. And in the end, as Dr. Sankaran referenced, the electron microscope made it possible to actually see these things. They are particles. They're just teeny, teeny, tiny particles. And in that sense, they might be soluble in the sense of not being like a precipitate or a cell, but they are actually particles. And so this duality between solution and particle turns out to be a false one. I mean, they're just tiny, tiny particles in solution. But I think that the kind of ontology of these things is very real, was made very dramatic by the electron microscope. For scientists who had other ways of detecting the virus, it might not have been so important, but for a wider public to see a picture of a virus makes it real.
This dual aspect which you meant, I think this also applies with Stanley. I mean, he had shown that the Tobacco Mosaic Virus is organic and inorganic by the crystallization process. I mean,
Exactly.
This aspect must have been completely confusing for several scientists around the globe.
Yes, absolutely so. And it continues a long history in biology of confounding phenomena which seem to be living and non-living at the same time. Whether it's is fermentation caused by a chemical or a cell? I'm trying to remember some of the other examples. I mean, for a long time, things were attributed to vital force because no one wanted to say that these living phenomena were caused just by chemistry and physics. But when you get to a virus, what'd you say?
Sorry, hormones was another when you were enzymes, hormones.
Hormones, exactly. So I think in this sense, and Stanley really played on this. The TMV was called it the twilight of life. It's like, is it alive? Is it not alive? It can't live on its own, but in the right condition and infecting a cell, it can reproduce itself. So it seems to be both alive and not alive at the same time. This invited another series of speculations as to whether viruses might be our evolutionary ancestors. Maybe things were kind of somewhat alive before they were fully alive. They're chemicals, but could reproduce themselves. But over the course of the 1930s, this theory was disproven, and instead viruses came to be seen as retrograde, evolutionarily retrograde, kind of stripped down forms of bacteria rather than as things that preceded bacteria. And to this day, I mean, much origin of life speculating now centers on RNAs, but figuring out what it was originally that was both kind of organic and inorganic, alive and not, remains an open question.
Even more than that, I think it's a very modern concept of a virus. I read recently that it may even influence the concept or the way the cell is programmed for its death in the tobacco plant. So I mean, a virus to dictate if a cell of the infected plant should die or not, or how to postpone this moment. I mean, that's something unique. I mean, some human beings will do everything in order to get the secret of the permanent youth, or however how to call it. So there might be, well, that's something very interesting. I mean, aging is something investigated by scientists, is something which keeps many people busy, something which belongs to life, but obviously for a plant infected by the Tobacco Mosaic Virus, it may work according to other rules. So aging in terms, at least for tobacco plants, seems to be more modern and sophisticated than for human beings.
That's a nice observation. And it does point to the fact that viruses co-evolve with the organisms they infect. And I mean, they're parasitic rather than symbiotic, but our attention now to the microbiome also shows that we're not alone in our life, in our evolutionary life, that we have co-evolved with many other organisms that can play as deep a role in our functioning as our own cells do.
And if I may add one thing to this, Andrei Loaf, I think it was, who pointed out, remember always that it is a cell that is the host of a virus, and the organism, and that cell is only part of the organism. So ultimately, and I think with plant cells, it gets a little more complex because plant cells are more, they're more, I know very little about plant cells, but I also know that they aren't as compartmental. They're not as cell-like as animal cells, especially human cells are. They have more chromosomes for one thing, and multiple ploidies, and they're a whole lot more complex than we would ever like to think. And so higher organisms sort of make that. One other point, Ivanovsky believed, I just want to go back to that, because while he believed it was a bacteria, what he believed was causing the disease was a toxin, because he was in that phase when, so he thought what came in the sap was the toxin, and that the bacteria were still too small, but that they weren't in the filter, right? I mean, they weren't being filtered, even though he found the filtered thing. It was the guys who did the Loeffler and Frush, who did, they're the ones who said it is particulate. So there was actually not a dichotomy, but there were three different things about how this disease could be caused. There were the people who said it was particulate, some people like Meyer and Ivanovsky more so, who said, well, there is a particle, but there's a toxin. And then Bayerink, who said, no, it's something completely new. And ultimately, there's, at some level, nowadays, if you think about it, it was a level of scale, like you said. And I thought that the level of scale is so important when we think about viruses.
Professor Creager, could you tell us, we have a few minutes left, something about Stanley, about the resistance which he met before he became famous, and then later on, and what about the difficulties and what helped him in order to implement also this type of model, and how did he influence further research of other disease?
He did a lot of important
work. I think when he made a claim that he had crystallized TMV, that was, as I mentioned, quite a sensational claim, in part because it made it seem like, oh my goodness, there's this pathogen that is as chemically simple as table salt. It's kind of a very mind-blowing idea. And of course, it wasn't that simple. It was a much, 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 much bigger molecule. But at the same time, the very fact that it was a molecule and not a cell or something even larger really did shift the ground for thinking about pathogens. So there was that level of resistance. He also, in his working with these very strong chemical reagents to precipitate the TMV as a crystal, he managed to kind of miss in his chemical analysis the fact that there was ribonucleic acid. He was only seeing protein when he did chemical analysis. And two British researchers, Peary and Bodden, who were actually working on a potato virus in England, decided to repeat his results with TMV. And they came from biochemistry and were more familiar. Stanley was an organic chemist. They were more familiar with the kind of repertoire of molecules or components that you might expect in a living system. And they knew more about nucleic acids. And they showed, in fact, that there is a small portion of this, which is nucleic acid, which is actually not, in this case, DNA, but RNA. Some viruses have RNA, not DNA. And so then, Stanley stood corrected about the constituent, the components of the virus, and it came to be called a nucleoprotein for a while. But it really wasn't until the 40s and into the 50s that people thought about these two parts of the virus as not just constituents, but related to one another, that the RNA actually encoded the protein that was this coat protein, as well as some other proteins that the virus uses to infect cells and multiply. And during the 1950s, as what's called the genetic coding problem came into focus, TMV was a very good model for trying to figure out at the precise chemical level how a change in RNA might specify a change in the coat protein. It was very, very laborious chemistry to try to figure out what the change in the RNA is and to sequence the coat protein to figure out where the change there was. But in principle, had not other systems been developed to do this more quickly, we might owe our genetic code to TMV. There was another attempt made with bacteriophage that was also very laborious. So that was another moment in which the system just kept on changing. You know, at first it's like, oh, it's just a crystal, it's just a protein. Then it's a nuclear protein. Then it's actually a little genetic virus, which has its own genes specifying its coat protein. And you know, over time, Stanley was very interested in how TMV could be used to understand human pathogens. In the 1950s, he was getting funding from the National Foundation for Infantile Paralysis, now known by its annual fundraiser, the March of Dimes. The organization is now just called the March of Dimes. And this was the organization in the US that lavishly funded virus research in order to understand what poliomyelitis virus was, and how a vaccine could be developed. And they funded the development of first the Salk vaccine and the Sabin vaccine. Well, obviously, once the vaccines were available, the Polio Foundation had no reason to put lots of money in virus research, but lots of virologists still wanted to get funding and solve practical problems. And the next thing that their attention landed on was cancer. There was, as Dr. Sankaran's book shows, already some long-standing viruses associated with cancer, Ralph sarcoma virus being the earliest and most important animal virus like this. And so Stanley really directed his laboratory then to look at cancer viruses, and many other laboratories and centers of research like Cold Spring Harbor Laboratory in the US were really oriented toward tumor viruses or cancer viruses. And what's interesting is that in the end, by the 1970s, there really weren't well-established links between these particular tumor viruses and human cancers. There are some with animal cancers, but not human cancers. However, this research on viruses had two unexpected benefits for biomedicine. One is that it led scientists like J. Michael Bishop and Harold Varmus to understand that the genes involved in cancers are on these viruses that are associated with cancer, but they're also in human cells. And this is the discovery of oncogenes, which all of us all of us have in our own bodies. They're not specific to viruses, as it turns out, but we discovered them because of the legacy of tumor viruses. The other is when the AIDS epidemic broke out in the 1980s, the very close attention to these tumor viruses meant there was a wealth of knowledge about animal viruses, and in particular, a kind of animal virus called a retrovirus. And that meant that that HIV, the virus associated with AIDS, was very quickly analyzed and understood to be a retrovirus. And although it took several years, that facilitated the development of therapies for AIDS. So this is all to say the identity of viruses kept on changing over this time, and so did the target diseases that scientists hoped to address. Sometimes they failed and sometimes they succeeded, but even when they failed, the knowledge itself was also beneficial.
And now, a few more words, Dr. Sankaran, if I may ask, what would have Behring have said that maybe 10 or 20 years later, scientists, in case of a severe pandemic, in case of COVID, may once more look at the Tobacco Mosaic Virus as a potential candidate to produce proteins which assembly the spike and could provide worldwide a new way of vaccines against SARS-CoV-2. I've spoken to Professor Lomonosov about this mechanism, how the Tobacco Mosaic Virus could produce the proteins for a future generation of vaccines against corona. What would have Behring have said that his discovery will be so relevant even 100 years later?
I think he would have been happy. He was, immensely happy when his ideas were brought up by Felix Darrell, one of the two co-discoverers of the bacteriophage, because Darrell got the Leeuwenhoek Prize and, Behring, and harked back to Behring, who was by then retired, and Byron had moved on and become much more famous as a microbiologist. This idea he always held on to, he had an affection for, but really his work progressed in other directions. I can't really speak for him. I just wanted to say Dr. Creager's work on cancer viruses has been underplayed here because, because she does model systems, she's able to jump from model to model in such a wonderful way. It's been really inspirational. So I had to say that.
Well, let's see what sort of models we'll see after this pandemic. What we know, I read a remarkable paper written by Professor Friedland but back to beginning of the years 2000, that humans have antibodies against a plant virus. And there's evidence that we have, smokers at least, have antibodies against the Tobacco Mosaic Virus. Robert Friedland, that's an article from the year 2003. Chinese scientists have observed during the pandemic, which I read in another article, that people with SARS-CoV-2 virus in the lung also had co-infection with the Tobacco Mosaic Virus. On the contrary, with patients who were infected with influenza 3 viruses, they had no traces whatever of the Tobacco Mosaic Virus. So it seems to me like that this Tobacco Mosaic Virus will keep us busy, and I hope for our best that it will have again a new contribution in maybe even understanding or treating COVID.
Can I just say that it's very interesting research. I haven't read these papers, and then I'll yield to Dr. Sankaran. It's important to stress that even when smokers get Tobacco Mosaic Virus in their bodies, it doesn't infect our cells because we're not closely related enough to the plants, which are its, hosts in the natural world. But it is true that we can have it in our bodies. So it's interesting to think about it being present as the viruses that actually do infect us come inside. But I wouldn't want anybody to worry. There was a concern in the age of research on cancer viruses that maybe TMV was a tumor virus. And in fact, it doesn't even infect human cells. So it's there's no need for smokers to worry about infected tobacco plants in this respect.
But maybe one of you might help me. I read a paper, and I'd like to quote a paper written by Professor Carrington, 1996, about the movement, virus movement involves specific viral and host factors. 1996. And I'd like to read one sentence. Additionally, movement defects of a virus or virus strain in a particular host can often be complemented by co-infection with an unrelated virus that is movement competent. Example, bromovirus, BMV, gains the ability to move through tomato if it is co-inoculated with the Tobacco Mosaic Virus. So if this coat protein has some similarities with, let's say, the proteins of another virus, could one imagine that the Tobacco Mosaic Virus would be like a key or a door opener for severe viruses for human beings? It's just a question which came spontaneously through my mind.
If I may, my idea with this, when one says antibodies against a virus, one has to realize that the antibodies are against a specific protein or a few specific proteins in the viral coat, which again goes back to what Angela said. Just because we can have antibodies to any protein that we're exposed to does not mean we're infected by the virus, because infection requires the virus to enter and multiply within our cells. That's not
co-infection
happening. Even with co-infection, co-infection happens. And what does it mean to co-infect? Even if it got into the human cell, unless it's going to multiply within the human cell and produce something, it's not causing any effect. It's just there, and it'll probably get out. So, I mean, I'm not saying this with a huge amount of authority, but we haven't learned enough to talk about jumping hosts that are so vastly different as animal and plants, have we, Dr. Creager?
Yeah, I share Dr. Sankaran's skepticism, but it's worth noting that the example you gave of the tomato plant, I totally believe that tobacco mosaic virus does infect tomatoes, and so do a variety of other viruses. Tobacco mosaic virus was one of the first viruses in which the ability of viruses to create ways to move between cells was well studied. There's a cell movement protein that enables the virus to move between cells. So that is one of TMV's strengths as a plant virus. So I totally believe that if it's infecting a plant cell and there's another virus infecting, that second virus might be able to kind of hitchhike, if you will, and go through the cells in the same way that TMV does. It's really hard for me to imagine a case in which TMV could have that kind of a role in human cells, because it's really kind of, if it's in a human cell, it's kind of an inert protein. It doesn't have kind of,
That's what
its RNA isn't going to result in the kind of proteins that will help it multiply, because the host machinery, as we would call it, the replicative machinery, and that virus's replicative machinery are just too far unrelated. They have to be unrelated to be able to sync up and work together. And that's what viruses do, is to exploit their host cell's machinery. So they're very, very specific to usually a small number of species. Now, with animals, I mean, we do see that the viruses can jump between animal populations, but that's because, especially like among mammals, we're very closely related. I don't think RSV, for instance, would ever infect a human, but RSV itself needs a helper virus in order to be able to cause cancer in chickens. So these relationships between viruses are very well established and well studied.
Professor Creager? Yeah, please, Dr. Sankaran.
No, when you said it's being used to produce antibodies, it almost struck me that are we using the tobacco plant as an antibody producing machine?
No, the author said that humans have antibodies against a plant virus, evidence from Tobacco Mosaic Virus. It's an article written by he's the corresponding author, Professor Robert, a neurologist, Friedland, and this is an article published in 2003. There's also research about mice, that the tobacco mosaic virus persists in the lungs of mice up to 15 days, and that it enters the mitochondrial cells. And there's also evidence that the tobacco mosaic virus may have an impact on the endoplasmic reticulum as well. So last but not least, when looking at the coat protein and the glutamic acids, what I have read in another article, we have no idea if these glutamic acids may not interfere with the NMDA receptors in one's brain. Surprisingly, 1919, and I love reading older medical publications. An author dealing with the encephalitis lethargica, he was not convinced that it had anything to deal, encephalitis lethargica, with influenza. And that's the last thing which I'll quote today. He said, the etiologic relationship of this disease to influenza rests on the above described coincidence of the epidemics. It is not common for the individual patient to have had a clinically distinct influenza. The encephalitis may itself be cerebral from form of influenza, or it may be caused by a separate virus, which in order to become active must have been in contact at one time or another with that of influenza. In none of the 11 cases which form the basis of the present paper had there been any different preceding influenza. So this is an article published by Peter Basso, I suppose Professor Dr. Peter Basso, in the Journal of the American Medical Association on the 5th of April, 1919. So this aspect of a possible co-infection with a tobacco virus, let's say, like Carrington explained, that it's like a door opener for other disease. This is an aspect which till end of April, I will debate with a plant pathologist, with a phytopathologist. I think that's the right name how to call this profession. neurophysiologist and an immunologist, because it's very interesting to see that we might have antibodies against this tobacco mosaic virus. The Pasteur Institute, the very famous Pasteur Institute in France, has observed that smokers made less severe forms of COVID than non-smokers. And it's also well known that smokers have better chances against the another autoimmune disease, the Parkinson disease. So some things relate to the tobacco mosaic virus, and I really will keep you updated and briefed with what we will find out in future related with this absolute magnificent and fascinating virus.
Well, I still think TMV doesn't have the right key to open up the cells of animals, but I look forward to your episode speaking with the neurologist and the plant pathologist.
It's just a curiosity. Professor Creager, thank you very much for joining us. That was a great honor interviewing you on the history of the research of the tobacco mosaic virus. Dr. Sankaran, thank you very much. It's late night in Bangalore. I really apologize for keeping you here in the interview for such a long time.
I'm fine. Yeah,
it's midnight. It's past midnight.
It's 12:30. No worries. I slept in the afternoon.
Good. Thank you for joining us.
Thank you very much. Have
really
a nice evening. Have a nice afternoon, and all the best. Ladies and gentlemen, thank you very much.
Thank you.
Bye-bye.
Bye-bye.